We're moving into the 5th chapter of section 3. This is, oh man, I can't wait till we get to the more complicated stuff. Well, thanks for answering my questions. Bye. See you next week. Reasons for TAing. Pays the bills, looks good on CV, the one at your undergrad who cares. I'll talk about this when we get to that particular chapter, but people that go into the sciences, you can't do a lot with a bachelor's. You can't do a lot with a 4-year degree. And so from your 4-year degree, you can either go into the training direction, which means medical school, vet school, dental school, all of which are going to be amazingly expensive and for which you're going to be paying for many, many, many years. But you're going to be making decent money most of the time. So, there's that balance. But for people that want to go on and get what's called an advanced degree in the sciences, a master's degree or a PhD, a doctorate, you go into a graduate program and they will pay you. They won't pay you much, but typically they will, your tuition is all covered, and some places your room and board is covered, and they do pay you a stipend, so they do pay you a salary. It's not a big salary, but it's a salary. And if you're not already paying huge student loan bills, it's probably enough to get by. I don't know, when I was in graduate school, my wife and I were together and she was working and I was TAing, teaching assistantship is what this is called. You can either get a laboratory assistantship where you work exclusively in the lab doing stuff, or teaching assistantship where they assign you to teach usually laboratory sections or freshman level sections. And that is another weird secret of universities, is very often if you're in the sciences in a university, you're being taught by people who barely have more education than you do. Is you're going for a 4-year degree and they just got a 4-year degree. Is that my first assignment, TAing, I was teaching an embryology class for people that were seniors. So I was a senior the year before, now I'm teaching seniors in a class where I had absolutely no background, had to learn it before I taught it. And also, when you're at the lower levels, at least people have, at least in theory, training in educational techniques. I've taken that training because I got certified to teach high school years ago, and I will tell you, it's largely a waste of time. It's terrible, involves the worst teachers you will ever run into. People that teaching are horrible. And I've heard enough people say that just not from my experience, it tends to be a very common response. Teaching educational psychology classes often are very useful, but teaching how to teaching is basically a performance art. Is that you can train people to some extent, but it takes a certain amount of leaning in a particular direction to get in front of a group and at least try to hold their interest for a while. So she's TAing. She's probably covering some class, covering a lab, writing exams, correcting papers. Teaching assistants do all sorts of things, in some cases with or close to total autonomy. Is that I went to graduate school in 2 different places. In one place, I met the professor teaching the lecture part of the lab maybe twice before the semester started, and maybe twice during the semester, and the rest of the time it's like, yeah, do that part, and just send me the marks. And then in another place is that we tended to be more like is that the professor would come in and go through stuff, and then we would wander around the class and make sure everybody was doing it right. So it's, again, very different approaches, very different places. I'll talk about that when we get to the chapter. I'm wasting too much time on it now. We're going to get into how cells interact with each other and with the world in general. So this is chapter 3.5. I know I don't always do this. I mean, it is in the label on the videos, so I don't feel like I always have to do that. And Cells can respond to information that they pick up from their surroundings. They are stimulated. It could be a chemical, it very often is a chemical. It could be light, it could be absence of light, it could be gravity, it could be a whole bunch of things. But the response can be positive, where the response produces a response in the direction of the stimulus. It could be negative, where the response involves a response away from the stimulus. And there's a number of different types. Some of them are so general that they just, a chemotaxis is fill in the chemical. Geotaxis is a response to up and down, is a response to gravity. Phototaxis is a response to light. Now it may be pure light, it may be a certain frequency of light. Again, it might be a positive, it might be a negative. Phototaxis is that plants tend to have a positive phototaxis where they turn toward the light. Whereas a lot of organisms in the water, you cast a shadow over them and they respond with a negative phototaxis. This very often involves receptors inside the cells or on the cells, usually to molecules of some sort or some sort of chemical. So most receptors have a reception site for some sort of chemical. It's not called an active site because it's not an enzyme. It's not causing a chemical reaction to happen most of the time. And we connected these to our senses. Sometimes the receptors are inside the cells. Obviously, you're going to respond to something that can get inside the cells if you have a receptor inside the cells. So a lot of light receptors are actually inside the cells. A lot of receptors for steroid hormones. Remember steroids? Lipid-based communication molecules. They tend to be able to move through a membrane because the membrane's lipid and they're lipids. They can go in, and the receptors for those are very often inside the cell. We'll talk about that in a little bit more detail. When there are things that attach to the receptors, they're called ligands or ligands. Remember, it's biology. If there's more than one way to pronounce a word, you'll find people pronouncing it every possible way. And they're often just pronouncing it the way that they were told it originally. And then you're sitting somewhere and you're listening to somebody going, "What word is that?" I've said this before. It's just, you get used to it. There are what are called signal ligands. Those are things like pheromones and hormones and alarmones. Neurotransmitters are a type of ligand, right? They travel across the synapse in order to connect to receptors on the other side. The control method is feedback. Something happens, you get a response. Usually the feedback is negative. So what? You get something and you get no response? No, the response is a response to control the message in a way that reduces it. Having said that, I just realized the example that's in my head is going to be even more confusing than most of my examples. But a thermostat is negative feedback. What turns the heat on? The temperature gets too low. So the temperature drops, the heat comes on, the temperature comes up, the heat goes off. Is that it's this you supply heat in order to counteract cold. Is that getting into this cold obviously kind of feels like a negative, but is that you've got something that can detect motion across a field, and it turns on a light, or it turns on your app that says, oh, somebody's come up to your door. But it's a negative feedback, is that the action produces something where you're going to respond to the action and hopefully make it go away. There is such a thing as positive feedback. Positive feedback is amplification, where the signal goes around and comes back in again bigger, and goes around and comes back in again bigger. The obvious example is that if you get a microphone too close to the speaker that the microphone is attached to, if there's a hum going through the system, the hum comes out the speaker, goes into the microphone, comes out louder, goes into the microphone, comes out louder, and eventually can overload the speaker, so it goes more. But there are positive feedback mechanisms in a number of multi-cell systems. A lot of our immune responses are positive feedback, where we get a small input and we have a large response. Inflammation, in fact, has to be controlled in our systems, because it can get, that's what an anaphylactic allergic reaction is it's an immune response that got too big and too out of control, way too fast, and your body wasn't able to throw anti-inflammatories, which you have a bunch of, at it and keep it tamped down so that it didn't kill us. Messages going into the cell and getting responses can be called transduction. This is a system that eukaryotes use, at least some variations of. There's a lot of variations of this. But let me get my little pointer. I'm going to leave this up here, so I'm not going to write a lot of this stuff down, so you're writing down as we go along, right? So here's your ligand. In this case, it's a hormone. And this is considered to be, in the system, the first messenger. It's bringing the message into the cell. And it attaches to the receptor. The receptor, in response to this attachment, changes shape. Now, this goes all the way through the membrane. It doesn't always. There are versions where the receptor's here, and then there's a communication molecule and another communication molecule, and it passes the signal through, one way or another. And one thing that's not shown here is on the inner space of the membrane is what's called a G protein. G proteins, there's a whole bunch of different ones. They're called G because they have guanine binding activity. And that makes the external signal internal. The G membrane will cause the release of a second messenger. And that's what this is. It isn't always cyclic AMP, but cyclic AMP is a very common second messenger. So it made sense for them to do this on the overhead as cyclic AMP. Cyclic GTP is sometimes used. Sometimes it's a release of something simple like calcium ions into the cell. It all depends on, this is a message. Keep in mind, the purpose of the message is to get a response down in here. It could be for the cell to do something, it could be for the cell to make something, it could be for the cell to make something so it can do something, but it's a response of some sort in the cytoplasm or possibly in the nucleus. We'll get to the nucleus part in a minute. Now, once we're in here, this may have a positive feedback loop where a small amount of first messenger causes a fairly large reaction because the second messengers are going round and round and round and round and just amplifying the signal. All depends on what the cell needs to do. There's usually thresholds. It's got to hit a certain amount of activity before it actually starts to do what you need it to do. Very typically, this sets up what's called a protein kinase. A kinase is an enzyme activator. So there's a step here that it's not really showing. There can be a protein kinase in here that actually turns the enzyme on. Remember, you have inhibitors to turn the enzymes off. Well, that means there's got to be times where you got to turn the enzyme back on. And the kinase may take the inhibitor away from the enzyme. Or hell, it may attach to another regulatory site and change the active site back to the right active site. All of this stuff is more complicated than the simple stuff we're describing, mostly. And a lot of times, this activation involves phosphorylation, right? ATP comes in, it's activated the cyclic AMP up here, but it also may activate the enzyme by sticking a phosphate group or transferring energy into the enzyme. This one is a little bit more complex. Here's your first messenger, your signal molecule. Is that the terminology for this can be a little bit all over the map. You've got your receptor protein, you've got your signal transduction pathway, so this is the G protein, and this is the kinase. You get a transcription factor. I'm not entirely sure when it's slightly out of focus. There we go. In this case, what's being activated isn't an enzyme, it's a transcription factor. Now, we haven't gotten to transcription yet. We'll get there in a couple of chapters. But transcription causes a part of the DNA, a gene, to be copied onto an RNA molecule so that you can make something with it, right? It's codes for proteins. Transcription factors will get that. Here's the RNA, it comes out and it makes the protein. So this is a signal that goes into the nucleus, gets the code out, and the response of the cell is to make a protein. Not to activate a protein that's already in there, but to make more of a particular protein. Maybe that's coming out of the cell, maybe it's going to cause the cell to do something. Who knows? Here's steroid, lipid-based hormone. This is not a barrier to that. The receptor is inside the cell. It may be inside the nucleus. This shows it as attaching here and then going into the nucleus. It's also not unusual for the lipid to go right in, and this is where the receptor is. And it activates a particular gene, and then the same steps, you get the protein to get made. Okay, that is part of the chapter. A lot of the rest of the chapter has to do with a lot of cellular interactions, including a lot of stuff about development. That'll be on the following video.